Hello everyone, welcome back to this series of lessons on chromatography. This is lesson 3, and in this lesson we're going to cover why some substances move further than others, and also what are the practical uses of chromatography. So before we begin, make sure as always that you've got your pen and paper handy. If not, pause this now and go and get them. Okay, so to begin with, let's warm our brains up again and try this retrieval task. So you've got 3 questions here. Which 2 colorants are contained in both sweet S and sweet P? How can you tell? Write the equation for calculating the RF value. And the solvent reached a height of 10 centimeters while colorant W reached a height of 3 centimeters. Calculate the RF value for colorant W. And all of this is using the diagram in the bottom corner of your screen. So have a go at those, pause this, have a go at those, come back and we'll go through the answers. Okay, so first question, which 2 colorants are contained in both sweet S and sweet P, and how can you tell? Well, if you have a look at the diagram at the bottom, what you want to do is look at the food colorants W, X, Y and Z, and you want to see if there's any spots W, X, Y or Z that match up to spots in both sweet S and sweet P. Well, if we have a look at W, there's got a spot in line with it at sweet S, and there's also one at sweet P, so that tells us that both of them contain W. X, there's no matching spots in either sweet for X. Y, we've got a matching spot in sweet S and a matching spot in sweet P. And then Z, we've got no matching spots in either sweet for that one. So the 2 that are contained in both sweets are W and Y. So the second one, write an equation for calculating the RF value. Well, from last lesson you should remember that RF is the distance moved by the spot divided by the distance moved by the solvent. And then let's put that into practice in question 3. The solvent reached a height of 10 centimeters while colorant W reached a height of 3 centimeters. Calculate the RF value for colorant W. Well, remember it's distance moved by spot divided by distance moved by the solvent. So you're going to do 3 divided by 10, which should give you a value of 0.3. A couple of things to point out here. Your RF value should always be less than 1. Okay, so that the spot can't move further than the solvent has. So you're always going to be dividing a smaller number by a larger number, so it should always be 1 or less. It's possible that your spot might move the full distance, so exactly the same distance as your solvent does, in which case your number will be 1. If it's more than 1, then you've made a mistake. You've probably put the numbers the wrong way around, so that's a quick way to check. If it's more than 1, just try flipping the numbers. The other thing I want to point out is that this RF value doesn't have a unit. RF values don't. Okay, we've done centimeters divided by centimeters, so we've cancelled the unit out. So it's just 0.3, no units. Okay, so let's get into it. Why do some spots move further than others? Well, the distance the spots moved is dependent on 2 factors. Those factors are how soluble the substance is in the solvent and how attracted the substance is to the paper. So there might be a force of attraction between the paper and the substance. Now, if that force of attraction is really strong, then the substance will tend not to move very far up the paper because it'll be attracted to the paper and stuck there, and the solvent will struggle to pick it up and carry it. So substances that have a high attraction to the paper tend not to move as far. But the bigger factor, the one that we're more concerned with, is how soluble the substance is in the solvent. So point A. Now, if your substance is really soluble in the solvent, then as the solvent comes up, it'll pick up your substance and it'll carry it a further distance. If it's not very soluble, then what will happen is it'll pick it up a little bit and it'll carry it a short distance, but then it'll drop it, and then some more solvent will come up behind it and pick it up again and carry it a little bit further and then drop it, and that process will be repeated until you stop the chromatography and take it out of the solvent. So as a result of that, very soluble substances tend to travel much further distances, and not very soluble substances tend to travel much shorter distances. So let's have a little checkpoint here. So let's pause, have a go at these questions, come back and we will go through the answers. Okay, so let's have a look at these. First question is, which food colorant is most soluble in the solvent, which is least soluble, and how can you tell? So looking at this diagram, we're looking at the food colorants, so W, X, Y and Z. We're going to ignore sweet S and sweet P at the sides, just those four in the middle. And what we can see is that X is the most soluble because it's moved the furthest distance. If you look at it, that spot is highest. So as the solvent has come up, it's picked up X and it's carried it quite far. So X is quite soluble in that solvent. Whereas Z, if we look at Z, Z's still right near the bottom, right by the start line. So the solvent's really struggled to carry that at all. So Z must be the least soluble in that solvent. And we'll know that because it's travelled the shortest distance. Question number 2, if a colorant stayed on the start line, what would that tell you? Well, if it stayed on the start line, let's think about what we said before about the 2 factors that determine how far a spot travels. If it stayed on the start line, then it must be because it's not soluble in the solvent that you've used. So if that's the case, then it won't get dissolved in that solvent and it won't move up the paper at all. And then question number 3, what could you change to allow that colorant to move up the paper? Well, you would try a different solvent. So it'll be soluble in something, so choose a different solvent and run the whole chromatography from the start again, but with that different solvent. Okay, so uses of chromatography, what do we use this for in real life? Well, if you think about the questions that are raised in your project, a major use of this is through the Food Standards Agency. Now, the job of the Food Standards Agency in this country is to make sure that food is safe for us to eat. So there's certain additives and certain colorants in foods that are banned, and they're banned for a very good reason. So sometimes it's because, in colorants in particular, it's because they've been linked to hyperactivity in children or something like that. And so the Food Standards Agency will do checks on different food manufacturers to make sure that they're using food colorants that are allowed and that aren't on the banned list. So they'll carry out chromatography and use that and compare it to the banned substances, and if the spots match, if they're in the same place, then they know that the food manufacturers are using colorants that they should not be. Another way that it can be used is testing for performance enhancing drugs in sports. So if you give a blood sample, if you're an athlete and you give a blood sample, then chromatography is one of the tests that could be used to identify whether your blood contains this banned substance. So again, you'd run the blood sample against a known banned substance and see if the 2 spots matched. Another interesting way that it's used is in testing inks. Now, yeah, that can be useful so that we can see what different colours make up an ink, but it's a really interesting use for that is in testing for forgeries in the art world. So certain inks didn't exist at certain points in history. So if you're testing to see if a piece of artwork is a forgery, then you can take a small sample of the ink and carry out chromatography, and if you find that it contains inks that had not been invented or discovered yet, then you know it's a forgery. It wasn't made in the time when it was supposed to have been made. And the last one I want to talk to you a little bit about is blood and urine samples. So if you give a blood or urine sample at the doctor's, then they may carry out chromatography on your blood or on your urine and compare that to known things if they're looking for specific things in your blood or urine. So an example is there's some diseases and disorders that cause your body to break down proteins into amino acids, and those amino acids appear in your urine. Now, in a healthy person, the body wouldn't be breaking down those proteins, so those amino acids wouldn't be in the urine. So they'll run chromatography on your urine sample, and they'll also do it alongside these known amino acids. And if they've got matching spots, then they can diagnose that you've got this condition where your body is breaking down the proteins that it shouldn't be. So those are a few uses of chromatography. There's actually loads, and what I'd suggest you do if you're interested in this is just have a little Google and have a read, because you'll come up with hundreds of different uses of chromatography in the real world. Alright, so let's see how well you've absorbed this information over the 3 lessons. The exam questions here are going to test your knowledge from across all 3 sessions, so make sure that you are up to date and that you've done them all in order before you try these exam questions. Now, as always, please make sure that you do write down your answers, don't just do them in your head. And this one is for 6 marks, so I'd give yourself about 6 minutes, pause it, come back, and we'll go through the answers and we'll see how you've done. Okay, so let's have a look at the answers for question number 1. So the question was, a recent study identified a possible harmful effect on children's nervous systems by some artificial colours. 2 of these colours are brilliant blue, which is E133, and quinoline yellow, which is E104. Both are artificial colours because they're made from coal. The company is to stop producing the blue sweets because it's removing all artificial colours and there is no natural blue alternative. So question A, suggest why it's important to be able to identify the colour additives in food. Well, the answer for that is to check that it's safe to eat. So to make sure that the colours that are being used aren't on the banned list, that they are permitted. So check that it's safe to eat or that it's healthy and that the colour is permitted. You can there also reference allergies and medical problems. So some people have specific allergies to certain colours, and so food labels have to have exactly the right colours written on them so people with those allergies can be sure not to eat them. So if you've mentioned allergies or medical problems, you would also get the mark for that. Okay, part B. A brown colour used in sweets was analysed using chromatography. The results were compared with those from E104 and E133. So remember, those are the 2 banned ones, the brilliant blue and the quinoline yellow from the article at the top. So the first picture shows you the chromatogram for E104. The middle picture shows you the chromatogram for E133, and then the third picture shows you the chromatogram for the brown colour. And the question is, what do the results tell you about the brown colour and its suitability for use in sweets? Now this is worth 3 marks, so be careful, make sure you're saying enough to get yourself 3 marks. You should be saying a mark per point. So the answers were any 3 from it's made up of 2 colours. So brown is made up of 2 colours, and we can tell that because there's 2 distinct dots on its chromatogram. It contains an unknown colour or an unknown dot. Now we can tell that because if we look at them, the top dot on the brown colour, the one that's travelled the furthest, that doesn't match E133 and it doesn't match E104, so that's an unknown. Thirdly, you can say that it definitely contains a harmful colour, and we know that because the first dot, so the lower down one on the brown colour, matches identically to E104. So that means that the brown colour contains E104, and we've established that that is harmful. You could then also say that it contains E104, or it contains quinoline yellow, or it does not contain E133 or brilliant blue, because there's no spot that matches to the E133. And then the last thing you could say is further analysis is needed, and I think that's an interesting point because we have discovered that it contains E104, but we haven't discovered what the second component is, so further analysis might be needed to decide if that's safe. Okay, so part C, once all the unsuitable colours are removed, the company claims that its sweets are now free from artificial colours. Does this mean that the sweets contain no additives? Explain your answer. Well, no, it doesn't mean that at all, because remember, additives can be all sorts of things. So you might put additives in and change the taste. You might put additives in and make it keep longer so it doesn't go stale or go off. Colours are only one type of additive, so there might be other types of additives in there. And the second thing is that they've said that it's free from artificial colours. So it might actually contain additional colours, but those might be natural, so they might come from nature rather than being artificial. So 2 things there. It could contain any number of other types of additives, and it could also contain different colours as long as those colours are natural. Okay, so give yourself a mark out of 6 for that one. Question 2. This one's only worth 2 marks, so pause this briefly, write down your answer, and then come back and check it, and we'll see how you've gotten on. Okay, so question number 2 was, chromatography was carried out on a sample of soft drinks to check that they contained only colours that were safe, and this is the result. So you've got the safe colours is on the narrow piece of chromatography paper, and then you've got the colours from the soft drinks, so A, B, C and D. What conclusions about the safety of colours in soft drinks A, B, C and D can be made from the results shown by chromatography? So it's worth 2 marks. So we can say that the drinks or the colours B and C are safe. So if we have a look at B and C, then the bottom spot of B matches with the bottom spot of the safe colours, and then the top spot for B matches with the third spot on the safe colours, so they're both safe. And then for C, the bottom spot of C matches with the second spot on the safe colours, and the top spot of C matches with the top spot on the safe colour, so they're both safe. So B and C are safe. We can also then say that colours A and D are not safe to drink. So if we have a look at colours A and D, our drinks A and D, the spots for A, or the first spot for A, doesn't match anything on the safe colours, which means that first spot isn't a safe colour. The second spot does, but the first one doesn't, so A isn't safe. And for D, the first spot doesn't match, but the second spot does, so that first spot is not something that's declared as a safe colour, which means that A and D are not safe to drink. And then finally we've got question number 3, so this one's worth 3 marks. Again, make sure you do write down your answer, pause this, come back, and we'll see how you've done. Okay, so question number 3 was, some farmed salmon have a colour additive in the food that they're given. This is permitted additive that improves the colour of the fish meat. Okay, so the food that is fed to the salmon contains this additive, and that then improves the colour of the meat, the meat on the salmon itself. So a sample of the colour is extracted from a salmon. Explain how paper chromatography could be used to confirm that this is the permitted additive. Okay, so you're making sure here that that is the additive that's allowed, it's not a different additive, a cheaper additive that they're adding instead. So explain how paper chromatography could be used. So we're going to use a solvent or solution or water or any named solvent for the first mark. So you're going to state the fact that you need a solvent. You're then going to say that that solvent carries or separates the colours or the dyes as they move or run up the paper. And then for the third mark, you're going to say that it matches against the RF value or a known chromatogram, or has the same pattern as the permitted additive, so it matches the permitted additive. Okay, so you're going to have run your chromatography on the sample that you took from the fish, and a second chromatography on the known additive, and you need to make sure those 2 chromatograms match up and have the same RF value. Okay, so that brings us to the end of lesson 3 in this series. So hopefully that's been helpful. Go back and have a look at the project again and see if you can add any detail using the information that you've learned from these. Thank you very much, and we'll see you back in September.